Hello, good afternoon, good morning, good afternoon, good evening. Welcome everyone to our last Global Immunotalk of the year. Global Immunotalks are a wonderful initiative created by Dr. Carla Rotlin and Lenina Zuniga in 2020 to spread immunology and inspire the new generation of immunologists all around the world in a friendly and egalitarian manner. I am Gabriel Rabinovich and have the honor and privilege of presenting our today's speaker, Dr. Jakub Abramson. Jakub Kobi Abramson was born in Prague in 1976. He earned his PhD in 2005 at the Weizmann Institute of Science. He then spent 5 years conducting postdoctoral research at Harvard Medical School with Diane Mattis and Christophe Benoit before rejoining the Weizmann Institute in 2011 as an independent group leader. He became a tenured professor of immunology at the Weizmann Institute in 2018. Since establishing his own lab at the Weizmann Institute, his research group has been mainly interested in understanding how immunological tolerance to self is established in thymus and how breakdown of this process results in autoimmunity. In addition to basic research in the fields of immunological tolerance and autoimmunity, the Abramson Lab also aims at translating their findings into novel tools for immunotherapy and or diagnostic of autoimmunity and cancer. His research has yielded several landmark studies which significantly contributed to deciphering the molecular and functional diversity of thymic epithelial cells and deciphering their functional roles in regulated thymus homeostasis and central tolerance. In addition to conducting research in the above-mentioned areas, Dr. Abramson is also very active in teaching several immunological courses. Moreover, Kobi was awarded a number of very prestigious awards and scholarships, including his selection into the Israel Young Academy in 2019, European Research Council Consolidator Grant Award in 2016, Kiel Prize for Excellence in Scientific Research 2016, and the Morashah Brain Drain Grant Award from the Israeli Science Foundation in 2011, among others. The title of Kobi Abramson today talk is Breath of Fresh Air. Air is aire, is in Spanish, so it's a very nice translation in Spanish, from broken central tolerance to broken Ts and antifungal immunity. So Jakub, as you know, one of the traditions of Global Immunotalks is to ask a special question before you start your talk to inspire the young generation of scientists. So the questions today are two interrelated questions. What challenges did you face when transitioning from trainee to independent researcher, and then what do you believe is the most critical skill for a scientist to develop early in their career? Please.
Thank you, Gabi, for the kind introduction, and thank you very much for inviting me. It's really a great honor to be speaking in this wonderful series. So actually, it's going to be 14 years since I have started my lab, very soon, in a few days. And I think I was really shocked by the transition because being a PhD student or a postdoc, it's kind of same, but when you transition, you suddenly realize that you need a whole toolbox of soft skills that nobody has told you. Nobody has taught you. So I think one of them is really like realizing you have to become a good HR manager in finding the right people and really identifying the qualities or potential problems in the people that you want to work with. You have to become a psychologist in guiding your students and solving problems of others. You have to become a politician in really trying to understand when to be smart and when to be right when negotiating with your colleagues. You have to be a teacher. You have to be a writer, a fundraiser. You have to be a marketer, a skill that I guess I'm going to exercise or try to exercise today. So I think it's a whole bunch of skills. And for me, I think the most critical one is probably the HR manager. So I hate bossing people. So my trick is really to choose students that are humble, modest, and know how to manage themselves. And then they save me time by managing them. So, and about really the skills, I think, again, it's the combination, but I think you cannot do science without curiosity, without creativity, and out of the box thinking. So I think this would be my picks.
Thanks so much. Thanks so much for the national.
It's really a pleasure to host you. And now I would like to invite you to share the screen and deliver your lecture.
Thank you very much. Just let me know if you see the screen.
Perfect.
Perfect. All right. So good morning, afternoon, and evening, everyone around the globe. So today I would like to tell you a story how we got from studying broken central tolerance in the thymus to study broken teeth and also broken anti-fungal response. And before I present you two separate stories, but related stories, I'll try to provide a brief introduction, which will give me a basis to introduce the main research interests of the lab and also the rationale for these two studies that I'm going to present today. So as I guess all of you know, T cells are really indispensable components of our immune system. We cannot live without T cells. We would not survive, or without T cells, we would survive only for a couple of weeks or months at the best. And this is best illustrated by two experiments of nature. One is a congenital or genetic disorder where a mutation in certain genes that block T cell development results in severe combined immunodeficiency, a disorder that really we cannot, is fatal unless it's treated. So today it's treated by bone marrow transplantation. And another good example is the AIDS disease or caused by the HIV virus, which gradually knocks down or eliminates T helper cells and basically knocks out our immune response. And again, we will acquire immunodeficiency, which eventually will be fatal. So the only place where the T cells can develop in our body is the thymus, which really has the amazing capacity to generate as broad a repertoire as possible of T cell reactivities that on one hand can recognize really an astronomical, or I would say even an unlimited number of pathogens and antigens that surround us, but at the same time have to be tolerant to thousands of self-antigens that are expressed in our body. So today we take this information for granted and kind of obvious, but this has not been the case actually till relatively recently, till 1961, when Jacques Miller, for the first time, described the functional significance of the thymus, which was till then regarded as a completely useless organ in our body. And Jacques Miller, as maybe not everybody knows, is also a person that has demarcated the difference between T lymphocytes and B lymphocytes in mammals, and still hasn't received a Nobel Prize, even though he's still alive. So I would like to stimulate this audience to push for his nomination for a Nobel Prize, which I think for his fundamental discoveries and contributions to immunology, he truly deserves. So the T cell developmental process in the thymus takes several weeks and is orchestrated by stromal cells of the thymus, namely the thymic epithelial cells, which we divide based on their anatomical localization into two major types: cortical thymic epithelial cells, or CTECs, and medullary thymic epithelial cells, or MTECs. And these two types of thymic epithelium, they are not only located at different sites of anatomical sites of the thymus, they are also characterized by unique and different functional capacity. So the CTECs are really critical for the early steps of T cell development. They provide signals for the recruitment of T cell precursors into the thymus. They provide signals for these precursors to commit to a T cell lineage. They also provide signals for the expansion of these developing T cells within the thymus. Now, they are also the sole mediators of positive selection of functional T cell clones, which have the capacity to recognize peptides in the context of MHC class I or MHC class II molecules. These positively selected clones then migrate to the medulla of the thymus, where they meet the medullary thymic epithelial cells, which are involved in the later steps of T cell development, including the negative selection of self-reactive clones and agonist selection of regulatory T cells. So the MTECs, they are not important only for the induction of immunological tolerance. They also represent a unique paradigm in biology, because unlike any other cell type in our body, these cells are equipped with the capacity to express about 90% of our genome. So every other cell type in our body would express between 50 to, let's say, 70% of our genome, with a large fraction of the genome being repressed. MTECs actually have the capacity to express up to 90%. And the reason for this is to really create a library of self-proteins, which are then presented on the surface of these MTECs in the context of MHC class I or MHC class II molecules to developing T cells. And if a potentially self-reactive T cell clone will see a self-peptide with high affinity, it will be clonally deleted, because such a clone would be dangerous and could cause autoimmunity, or would be converted into a good cop, which is FOXP3 regulatory T cell, which then maintains immune homeostasis and tolerance in the periphery. And even more strikingly, about 5,000 genes, which is a quarter of the genome, is induced by a single factor called the autoimmune regulator, or AIRE, a transcription regulator, which in a very sophisticated manner manages to turn on genes that are normally repressed in most of the tissues. We call these genes tissue-specific antigens. An example would be, for example, insulin, which no cell in our body would express except for the beta cell in the pancreas, and MTECs in the thymus. And to really illustrate the functional significance of this process and of AIRE, I think it's best to look at experiment of nature, which is a very devastating autoimmune syndrome called APS1 or APSET, caused by the loss of function of the AIRE gene, which, as you would expect, results in the development of a whole plethora of autoimmune diseases. A patient with AIRE deficiency might develop up to 20 different autoimmune diseases during their lifetime, including Addison's disease, hypoparathyroidism, vitiligo, type 1 diabetes, you name it. Okay? This disorder is also characterized by two peculiar phenotypes, which is chronic candida infection and enamel dystrophy. And today I am going to focus on these two peculiar phenotypes and trying to understand, tell you the story, how we try to understand the mechanisms why AIRE deficiency results in these interesting phenotypes. So the research in my lab is mainly centered around three things: the thymus, the AIRE gene, and autoimmunity. And while in the beginning of my career, we were really interested mainly in the overlapping parts, More recently, we are kind of also looking at the non-overlapping part. So we are really interested in the thymus biology, especially the tech biology, understanding the diversity of the thymic epithelial cells, because it's not as simple. The MTech, for example, they are not a homogeneous population, but a vastly heterogeneous population. We and others have demonstrated that actually there are many, many different cell types that are expressed in the thymus and comprise the MTech compartment, which we call MTech mimetics. We are trying to understand the role of air in the induction of central tolerance, and also how its dysfunction results in breakdown of tolerance and autoimmunity. More recently, we got interested in extrathymic air-expressing cells that are present in the lymph nodes and express the ROR-gamma T transcription factors. And I'm going to talk about these cells in the second half of my project. We are interested, obviously, in general in autoimmunity and exploiting autoimmunity for cancer immunotherapy, specifically by cloning autoantibodies that have very high tissue specificity and tumor specificity, and exploiting them for immunotherapy purposes. And more recently, we got also interested in the role of sex hormones in autoimmunity. So today I'm gonna focus solely on air and tell you one story about the air and the loss of air in the thymus, and another story in the second half, the loss of air in the extrathymic air-expressing cells. So as I mentioned already, there are two very peculiar phenotypes that characterize most of the APS1 patients, the air-deficient patients. And one of them is this enamel dystrophy. About 90% of air-deficient patients will develop enamel dysplasia. And the reasons for this were completely unknown. And this really intrigued us, especially my former PhD student Yael Groper, who really tried to understand why enamel dystrophy, which doesn't look like an autoimmune disease, is exhibited in air-deficient patients. So this project was done in a close collaboration with several labs around the globe, including Øystein Husseby's lab in Norway, and Hannes Wolf's lab, Jan Prochaska lab in Czech Republic, Karol Gelineau in France, and Susa Sondi in Hungary. So before I jump into the results, I have to introduce the process of forming the enamel layer, which is the hardest tissue in our body, the process of amelogenesis. We are immunologists, and I personally knew very little, almost nothing about this process. So the enamel is built. Imagine it like you want to build a wall. First, you need to create a scaffold of proteins that are secreted by a unique epithelial cell type that is in our gums called the ameloblast. These cells secrete several unique proteins, which are tissue-specific antigens. They are virtually expressed or mainly expressed only by these ameloblasts, called, for example, ameloblastin, ODAM, enemalin, or amelex. And these form a scaffold onto which crystals of hydroxyapatite are deposited. And once these crystals are deposited and we generate this enamel layer, the scaffold is then dismantled by proteolytic enzymes like MMP20 or KLK4. And it has been shown that actually there is a disease that kind of resembles the phenotype, the enamel phenotype in air-deficient patients, and this disease is called amelogenesis imperfecta. Is a genetic disorder which can be caused by loss of function in about 20 different genes, but virtually all of these genes that are listed in this table are kind of part of this amelogenesis process. And most of them are proteins that either make the scaffold or disassemble the scaffold. And loss of function of any of these proteins will result in enamel dystrophy, very similar to what we have seen in the APS-1 patients. So when we started this project, we had two main hypotheses, a developmental hypothesis, where we hypothesized that maybe AIRE might be expressed extrathymically in the ameloblast and somehow control the expression of these proteins in the enamel matrix. The other hypothesis was an autoimmune hypothesis and we hypothesized that these unique scaffold proteins are expressed actually by AIRE in the thymus, and we generate tolerance through AIRE to these proteins. And when AIRE is not there, actually the tolerance is broken down, and we generate either autoantibodies or self-reactive T cells that would destruct the enamel layer. So to cut this story short, we very soon could disregard or disprove this hypothesis. We didn't find any evidence that AIRE is being expressed in ameloblasts, or that would control the expression of the proteins in ameloblasts, which kind of left us to test the other hypothesis, which was the autoimmune hypothesis. And this hypothesis was really supported by a lot of data. First of all, when we looked at the expression of enamel-specific genes, either in AIRE wild-type mice, in MTEX isolated from AIRE wild-type mice, or isolated from AIRE-deficient mice, you could see that many of these proteins were simply not expressed or downregulated in AIRE deficiency, including these in the red, which I highlight. All these genes have been previously linked with amelogenesis imperfecta, suggesting that these AIRE-deficient mice might develop breakdown of tolerance to these proteins. So to test this further, we isolated sera from either AIRE-deficient mice or on NOD background or wild-type mice, and also isolated ameloblasts and make cell lysates from these ameloblasts, mouse ameloblasts, and probed these ameloblast cell lysates with the sera from individual AIRE-deficient or AIRE wild-type mice. And each lane, what you see here, is a serum from a different either AIRE-deficient or wild-type mice. And what you can see here, that you see a lot of reactivities for sera that is isolated from AIRE-deficient mice, while there is very little reactivity for sera that are isolated from AIRE wild-type mice, suggesting that the sera is reactive to ameloblast proteins. And to really figure out which proteins are affected, we expressed many of these ameloblast-specific proteins as recombinant proteins and tested them in ELISA, and I'm showing you a few examples. Reactivity to amelogenin, to ameloblastin, and ACPT. You can see that in these cases, you always see much higher reactivity for sera that is isolated from AIRE-deficient mice, and very little reactivity from mice that are wild type for AIRE. When you look at the teeth of the AIRE-deficient mice by naked eye, you hardly see any phenotype. Sometimes you see that the teeth would be a bit whiter, but I don't think we could make any kind of statistical confirmation for that. So we decided to actually do a more sophisticated analysis using micro-CT. And when we scanned the teeth from either AIRE wild type or AIRE knockout mice, we noticed that actually many, not all, but many, maybe more than half of the AIRE-deficient mice had these pits in the enamel. You can see it right here, which are kind of reminiscent to the same defects you see in the AIRE-deficient patients. Another way to look at this is really to look at enamel or enamel density or area along the tooth, along the incisor, and compare the density between the left and the right tooth for each mouse. So if you look at the wild type, you see the density is quite homogeneous for the left and right teeth. But it's kind of scrambled in air deficient mice. So for example, here, the left tooth is kind of affected. There is very little enamel while the right tooth is not affected. It's kind of characteristic also for the human APS1 patients that one tooth would, on one side would be affected and the corresponding tooth on the other side would not. So another thing you could really visualize and get impressed by the phenotype is to do electron microscopy. So these are beautiful enamel crystals on from a wild type mouse. You really see these beautiful crystals, and the affected area on the air knockout mice, it really looks like a scrambled egg. See, really the crystals are there, but they don't have this beautiful, beautiful structure. Excuse me. So these were results from mice, and what about humans? So we spent like 2 years trying to show that there is reactivity also in the human sera, and we were failing because we were always looking for IgG reactivity, and we couldn't find any differences except for one protein, ACPT, which had IgG reactivity. Till we realized that maybe we should also look at other antibody isotypes, especially IgA, which is also T-cell dependent isotype. And in this case, we saw very significant differences in the autoreactivity to number of proteins. You could maybe see it better in this graph, in this heat map. So red means reactivity for the individual protein in APS1 patients or in control patients. So there is quite a lot of reactivity to any and kind of, you see that certain patients would have reactivity to more than one such proteins. And another way that we looked at these data was that we took children aged 4 to 14 years old and segregated them by their enamel phenotype into either a mild enamel phenotype or a very severe phenotype, and looked at the reactivity of their sera to different enamel proteins. And you can again appreciate that those children with a strong phenotype had much higher reactivity than those that with a mild phenotype, suggesting, again, this is not causality, it's just correlation, but suggesting that the higher reactivity, the stronger the phenotype they may develop. So this helped us to actually introduce a brand new autoimmune disorder that has not been described before, which we termed autoimmune amelogenesis imperfecta. And I think using these APS1 patients was really essential or critical for kind of discovering this new type of autoimmune disease which might have been overlooked. Now, the story doesn't end here because air deficient patients, all these autoimmune disorders they develop, they also exist or in patients that have wild type AIRE, that have functional AIRE, and it might not be a result of AIRE deficiency. And also, many autoimmune disorders often occur together. For example, thyroid disease often occurs together with vitiligo. So we ask, maybe this is this autoimmune disorder is much more common than we think, and it's not only restricted to APS1 patients and might exist also in other patients. So we looked, and actually it was Eistan Husabi's idea, who suggested that actually celiac patients are characterized by a similar enamel dystrophy. And I haven't know about this, but this is actually something that has been known from the 1990s, that a large fraction of celiac children will develop a similar enamel dystrophy. So we looked also at autoreactivity to the same panel of recombinant proteins we had from non-celiac patients and celiac patients, and saw a very similar phenotype. Again, the reactivity was mainly mediated by IgA autoantibodies, and a similar table, similar heat map here showing that you see much higher reactivity to many of these enamel-specific proteins in celiac versus non-celiac patients. So as you know, celiac disease is an autoimmune disease that has an environmental trigger, specifically a gliadin, a protein that is part of wheat. After digestion, it has to be deaminated by transglutaminase. And in celiac patients, because probably of the leaky gut, the transglutaminase becomes a target of the immune system, where the immune system starts to generate autoantibodies against this enzyme, resulting in eventually in an autoimmune phenotype, an autoinflammatory phenotype. So the obvious thing was whether it's because of the TGM. So we looked at TGM deficient mice and actually saw very mild enamel phenotype, suggesting that at least in mice, the mechanism might be different. Again, in humans, it could be the TGM, but at least in mice, it seemed like a different protein. So we hypothesized maybe we should look again at the list of the 20 so proteins that were linked previously to amelogenesis imperfecta, and ask maybe any of these proteins might be expressed both in the enamel and in the intestine. And maybe there is a cross-reactivity. Maybe the celiac patients will develop autoreactivity against this protein in the intestine, and then the autoantibodies might interfere also in the enamel formation in the celiac children. And by screening all these proteins, we found basically only one candidate, which is LAMP3, laminin B3, which is, as you can see here, very specifically expressed in the enamel layer, but it's also highly expressed in the intestine. And we have shown that celiac patients develop autoantibodies, IgA autoantibodies against the LAMP3 compared to the healthy patients. So again, this is not causality, it's just a correlation, and the LAMP3 might be one of such antigens that is expressed both in the intestine and in the enamel, and to which celiac patients develop autoantibodies. But there is another mechanism that we were absolutely fascinated by, and we came across this by actually looking at the ameloblast locus, where we realized that the locus that encodes the ameloblast-specific genes is located both in mice and in humans just next to another locus encoding for casein genes, okay, including kappa-casein. And by looking at our transcriptomics data, we actually noticed that the casein-3 gene, encoding kappa-casein, is highly specific also for ameloblasts. And you can see it here by microscopy that it's kind of co-localized with amelogenin. So kappa-casein is an ameloblast-specific gene, in addition being expressed in the mammary gland and being part of the milk. It's a milk protein. So since caseins are abundant proteins that are present in milk, we hypothesize that maybe similar to gluten, there might be another common food antigen, namely kappa-casein, that most of us encounter on a daily basis. Might be kind of breaking our immune tolerance in celiac patients. So, and this was further supported by actually the fact that the kappa-casein had kind of a similar evolution like gluten, because as you know, the amount of gluten in wheat increased in the past 50 years, maybe by order of magnitude. So the concentration of kappa casein in milk, and the reason for this is that the kappa casein is absolutely essential for cheese making. So there is a lot of inbreeding and selection for cows with high kappa casein yield, so that making cheese will be more economical. So that was also interesting. So what we asked was whether celiac patients might have higher reactivity to bovine kappa casein, and the answer is yes. So you can see here again quite a strong reactivity of IgA antibodies of celiac patients to kappa casein from cows. You see some reactivity also in healthy patients. And it's mainly the IgA, only in few patients it was IgG1. The bovine kappa casein and the human kappa casein are mildly conserved, but there are certain epitopes that are highly conserved. And actually, we found that there is a cross-reactivity against these epitopes. The best way to illustrate this is here. You can see all patients, celiac patients that had high reactivity to cow kappa casein had also high reactivity to human kappa casein. So not everybody that had high reactivity to cow kappa casein cross-reacted with the human one, but as you can see, none of those that had a low reactivity cross-reacted. So we believe that this reactivity to the cow milk or cow kappa casein might be maybe another reason for a trigger for this very peculiar phenotype. So to sum it up, this part is that in APS1 patients, the mechanism is very, very simple. Without AIRE, you don't have expression of a battery of enamel-specific genes in the thymus. And as a result of this, you develop a breakdown of immune tolerance to these antigens, which then manifests as this autoimmune amelogenesis imperfecta. In celiac patients, the mechanism, I think, it's much more complicated, and I don't think we understand it fully, but we propose that it's mainly breakdown of peripheral tolerance because of a leaky gut and exposure to either some common food antigens like kappa casein or expression of genes like LAMP3 that are part of the intestine and are also very important for the amelogenesis. And again, I want to stress this has to occur only during the phase of amelogenesis in children, meaning after birth till the age of 6. If you develop these autoantibodies later on, it then doesn't really matter because the enamel has mineralized and the autoantibodies cannot interfere. So this might be kind of solved. And there was another intriguing phenotype that interested us. And this was how and why APS1 or the AIRE deficient patients develop this chronic candidiasis. So about 90% of AIRE deficient patients would develop this phenotype. And actually, this would be the earliest phenotype you will usually observe in these patients. So this question has been addressed by my former postdoc, Jan Dobesh. And again, before I show you some results, I want to explain a little bit about the mechanism, how our body copes with candida infection, which is really mediated by our solely, like the key player are Th17 cells. So the candida overgrowth on mucosal tissues gets disseminated. The current understanding is that it's taken up by phagocytes like dendritic cells, which then bring it to lymph nodes, prime T cells, and drive their differentiation into Th17 cells, which then migrate back to the infected tissue and provide help to the infected epithelium to produce all kinds of defensins and mucins, which suppress the candida overgrowth. So, and this is really best illustrated by, again, several experiments of nature, human patients that are born with deficiency in a number of genes that are key in the development or for the development of the Th17 response. So some 14 years ago, two back-to-back papers were published in JXMed that actually suggested that also the candida infection in APS1 patients has an autoimmune context, namely that the APS1 patients develop autoantibodies against IL-17 and IL-22, which are neutralizing and then basically depriving the help that Th17 cells provide to these infected epithelial cells. So this is a very elegant hypothesis, and I really like it, and I think to a large extent it is true. But if you really look at the data, there are actually several things that might suggest that there might be some additional mechanisms. So not all APS1 patients that get these antibodies develop chronic candida infection, and vice versa. So maybe there is also something else. So again, we went to study this in mouse models using AIRE-deficient mice. And we did a very simple experiment. So if you infect wild-type mice by either live or heat-killed candida, you will see for a few days a very nice Th17 response by measuring ROR gamma positive T cell proliferation. But when we did the very same experiment in AIRE-deficient mice, we saw almost no Th17 response. So that was kind of surprising, suggesting that there must be an impact on Th17 cells. We also immunized either wild-type or AIRE-deficient mice by heat-killed candida, and then after 3 weeks infected them IV by live candida albicans and looked at their survival. And you can see that while about 50% of the wild-type mice survived, the AIRE-deficient mice succumbed in the first 10 days, or 100% of them, suggesting that there is a real defect in adaptive immunity. And this defect, at least in the B6 mice, was not mediated by these neutralizing autoantibodies, because the AIRE-deficient mice on B6 background don't develop neither IL-17 or IL-25 autoantibodies. And we saw a very similar effect also in APS1 patients when we isolated peripheral blood lymphocytes and stimulated them by candida and measured either IL-17 secretion or Th17 proliferation. There was again a very significant defect in the Th17 response, also in the APS1 patients. But you might now ask, why would defect in thymic AIRE result in impaired Th17 response? The Th17 cells are not generated in the thymus, but in the periphery. So this suggested to us that maybe extrathymic AIRE is involved. And indeed, even the first paper that cloned AIRE reported that in addition to the thymus, AIRE is also expressed in the lymph nodes to a quite high level. But since people got really excited about AIRE's role in immune tolerance, I think this role of peripheral AIRE has been overlooked. And really, thanks to Mark Anderson and Jay Gardner's work, I think they really pushed the research in this direction, and also focused on this extrathymic AIRE. But for me, the main breakthrough came in 2019 when Jan Dobesh, my former postdoc in his previous lab, described that these extrathymic AIRE expressing cells also express the transcription factor ROR gamma T. So back then, we thought that these cells are ILC3-like antigen presenting cells. Today, we know that they are not. So we call them ROR gamma T positive extrathymic AIRE expressing cells, or ROR gamma T ETECs. And we hypothesized that since they are antigen presenting cells, and they also have express high levels of CD80, CD86 molecules, that maybe these cells might somehow instruct CD4 T cells and program them towards the Th17 fate. So first, when we looked at the transcriptome of these ROR-gamma T positive ETECs, we noticed that they are equipped by several sensors for candida, like galectin-3 and dectin-1, which can sense candida and signal. We also did an experiment where we isolated these cells and ex vivo incubated them with the fluorescent labeled candida. You can see after 30 minutes, the cells try to make contact with the candida, and after 90 minutes, the candida is fully engulfed. We also did an experiment where we infected mice with candida, isolated several different antigen-presenting cells, and then the candida had ova antigen, and then in an ex vivo reporter system measured their capacity to present the ova antigen to T cells. And actually, while in the first days, the conventional antigen-presenting cells, like dendritic cells, dominated the response, after 3 days, these ROR-gamma positive ETECs were the most potent antigen-presenting cell for the candida antigens in the lymph node. So to study the role of AIRE in these cells in a more specific manner, we developed a conditional knockout using the ROR-CRE airflux mice. And I can show you here that when you measure the AIRE expression, the AIRE expression is beautifully deleted in this model in these ROR-gamma AIRE expressing cells without affecting AIRE expression in the thymus. So these mice retain expression of AIRE in the thymus, but they lack expression of AIRE in the lymph nodes. So we then infected these mice again with this systemic candida challenge, immunized them with heat kill candida, and after 3 weeks, injected them with live candida. You can see kind of recapitulates the AIRE full knockout. These mice did not survive, most of them beyond 10 days. And also there was a much higher overgrowth of candida in the kidneys. We did another model, which was oral administration of candida and monitoring the overgrowth in mucosal tissues, and also measuring the candida-specific T cells by tetramers that are specific to one of the candida proteins, ALS1. So you can see that the immunization in wild-type mice, you generate 300 ALS1 positive Th17 cells in the wild-type mice, but hardly any in the ROR-gamma driven knockout. And this is mirrored by an overgrowth of candida in the intestinal tissue in these specific knockouts. So what we are proposing is that AIRE has another job in the periphery, which is very different from the thymus, and that these ROR-gamma T-positive ETECs are very potent phagocytes of candida and actually help to expand the Th17 cells in the lymph nodes. And without them, the Th17 cells don't get expanded, and we have a defective response against candida. So another question we wondered, and I guess many of you wonder, is whether the expression of AIRE in these ROR-gamma T ETECs is critical for the induction or maintenance of tolerance to self, like the expression of AIRE in the thymuses. So it has been proposed that these cells might be very important for tolerance induction. So we did actually several models. One of them was generating a non-obese diabetic mouse with RORC-CRE AIRE deficiency. And so these mice should be, if AIRE plays a role in immune tolerance, these mice are very prone to develop diabetes and other autoimmune diseases. But actually, when we knocked out AIRE in ROR gamma T positive cells, ETEX, we didn't find any actually big impact on the development of diabetes or survival. In fact, the mice survived better and developed less diabetes. And also, when we looked at other peripheral tissues, I'm showing here a salivary gland. Here is reactivity. If you knock out AIRE in the thymus, you generate a lot of autoantibodies that are specific to the salivary gland. Nothing really happens if you knock out AIRE in the ROR gamma T ETEX. And another model we used is a model for arthritis, again, using this RORyt. And again, we didn't find any differences either in joint swelling or in the autoantibody generation in the RORyt positive or RORyt negative KBXN mice. So you wonder probably what are these ROR gamma T positive ETEX? When we are doing our studies, we were really kind of convinced they are ILC3-like cells, but then a whole bunch of studies just before our paper was published or just slightly after our paper was published appeared and characterized these cells using single-cell RNA sequencing and named them by alternative names, including the Janus cells or the Tethys cells. And for us, actually, was very important to characterize them in more detail. So we developed a reporter mouse using ZS Green reporter, where the ZS Green reporter is turned on only where functional AIRE protein is being expressed. So this reporter works really beautifully. And when we sort these ZS Green positive cells or ZS Green negative cells, like other antigen presenting cells, you can really appreciate that these ROR gamma T ETEX are a very distinct population, very different from ILC3s, from conventional dendritic cells. They really represent, I think, a novel lineage, which is still about to be characterized further. So what I just want to finish with is that they are not a homogeneous population when we actually focus on these cells, because we have single cell analysis based on many thousands of these cells. We can distinguish at least 5 different subsets highlighted here by these different colors. Each subset will be marked by a unique gene signature and surface markers. And when you actually, these cells, cell subsets seem to have very different functional roles. So for example, when you do Go analysis, so this EpCAM positive ROR gamma T ETEX will be predicted to regulate T cell migration and be involved in negative regulation of the immune system, while these CLEC4 positive ROR gamma T ETEX, actually they are predicted to play a role in anti-fungal innate immune response. So we believe these are the cells actually important for the anti-candida response. But there is another relatively big subset, which is defined by expression of a lot of neural genes and synaptic genes. And these cells are predicted to actually be involved in neuron projection development, suggesting that there might be a crosstalk between these cells and neurons innervating the lymph node. So the last slide I'm going to show you, and the take-home message is that AIRE plays very different roles in the thymus and in the lymph node. So in the thymus, it is absolutely critical for the induction of tolerance to self. While in the lymph node, we have shown that AIRE deficiency results in impaired generation of candida-specific Th17 cells. But these ROR gamma T ETEX, also known by other names like Tethys cells and Janus cells, might and have been implicated in additional roles, including induction of tolerance to harmless non-self antigens like microbiota and food antigens, And also, I think they might be involved in the crosstalk with the nervous system, so the neuroimmune crosstalk. So we are not focusing on this because too many labs are already working on this, but we are very interested also in this question. So with that, I'm going to finish and thank all our collaborators and mainly highlight Dial Druper, who was spearheading the enamel story, and Dian Dobesh, who spearheaded the candida story. So thank you for your attention and for joining today.
Oh, thanks so much. Thanks so much, Kobe, for sharing this wonderful work and this very exciting adventure. I think that the most important thing I see here is very creative work out of the box. So thanks so much and congratulations for your work. I'm sure there are plenty of virtual applauses. So as usual, this talk for everyone will be followed by questions and answers in Twitter, X. So please follow @GlobalImmuno, #GlobalImmuno. And finally, personally, I would like to express my sincere thanks to everyone on behalf of the organizers. Especially, I would like to thank Elina and Carla for leading this wonderful initiative, and wish you, everyone, a very happy, healthy, and successful New Year. And probably Elina will join now to say the final. Is Elina here?
I'm here, Gabi. Thank you so much.
Great.
Thank you, Jakub, for presenting and sharing your findings on Global IMMUNOTALKS, and thank you, Gabi, for hosting him. So I'm here to wrap this year of success in Global IMMUNOTALKS with really full of gratitude, right? So I would like to thank all the 2024 Global Immuno speakers who, through their super valuable contributions, have enhanced the general knowledge of immunology worldwide. So thank you all 2024 Global Immuno speakers for your efforts and for sharing your perspective here at this forum. And I would also like to thank the 2024 organizers, including you, Gabi, right? And especially Dietmar Sen and Kazuyo Moro, who have served for 2 years in 2023 and 2024. And we are not letting them go. They will continue serving Global IMMUNOTALKS as members of the advisory board. And of course, the rest of the 2024 organizers, Miriam, Caetano, Asha, Bali, and Gabi, right, who have agreed to continue serving as 2025 organizers, and will be joined by 2 new organizers that I think that you can see here, Ana Maria Lennon, and from the Curie Institute, and Hai Ki from Tsinghua University. And so this team of 2025 organizers have already made efforts to prepare a fabulous line of speakers for 2025. And so we also want to thank these speakers, and we cannot wait to learn from you. I think that these 2025 years of Global IMMUNOTALKS will continue being very, very strong. And we're looking forward to the incoming Global IMMUNOTALKS in 2025. And so to the audience, I'm also very appreciative, and we hope you continue joining us through in the next year, and that you continue helping us to spread the word. So we will be sharing this flyer in social media, X, YouTube, etc. And so we would really appreciate that you come forward and spread the word. And that's it. I wish everyone a wonderful 2025, full of peace and lots of learning. And so thank you again for joining us, and hope we can count with your attendance again in 2025. Thank you so much. Bye. Take care. Thank you, Jacob, again, and Gabi. Bye-bye.
Happy New Year.
Bye.